Have you ever asked yourself, how does DNA replicate? Is when a DNA molecule would get copied and make up a second new one. The second one, semi-conservative, is when a new strand comes with an old parent strand. Dispersive is when a DNA molecule will be cut into various parts, each of which would get copied and reattached to produce two DNA molecules. The third, the scientists didn't know. The scientists didn't know which of these theories is correct. The two scientists, Mathaway and Franklin, uh, had an idea to design an experiment. In May 1958, Franklin and Mathaway used uh, used a bacteria because it was easy to be grown in laboratory. They grow the cell with a specific type of heavy nitrogen and a low nitrogen. Eventually, it happens to be equal. This shows that the high nitrogen is mixed up with low nitrogen. And then they discovered that a new strand of low nitrogen is convert with a new high nitrogen, and that they found out the semi-conservative was the right one.
The first DNA replication is to separate the two strands with an enzyme called helicase. The separated strands each provide a template for creating a new strand of DNA. An enzyme called polymerase binds to the primer and will make a new strand of DNA. DNA polymerase can only add DNA bases in one direction, and one direction only. One of the new DNA strands, which is the leading strand, is made continuously. The DNA polymerase adds bases one by one.
The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. Then polymers can therefore only make this strand in a series of small chunks called Okazaki fragments. DNA polymers then added a short row of bases from 5' to the 3' direction. The next DNA polymers is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated. Finally, the enzyme DNA ligase seals up the fragment of DNA. Then a new DNA has been made.
And I'm here to talk about the linking. So first off, we start off with the original parent strand, where all of it is original. Then we move on to the next replication, which is the first one. There will be two sets of DNA, and it will become one original parent strand and one new parent strand. Then we move on to the second replication, which will consist of two fully new strands and two sets of one old and one new. Let's take the set with the new strands, for example. The new strands represent New Kuwait, also known as modern day Kuwait. And the old strand represents old Kuwait, like before the war. And we can see how the old Kuwait strands gets diminished. The replication represents how Kuwait as a society and as a country develops and change over time.